Welcome back, everyone. Hopefully, you got your head around using GatherTown just there. This is the second half of the first session. So I'd like to start this by welcoming our first speaker for the second half of the first session, who's an expert in the field of waterborne pathogen detection, here with us today to discuss the development of sensors for antimicrobial resistance. From Heriot-Watt University, please welcome Dr. Helen Bridle.
Thank you very much. Good morning, everyone. And nice to meet you all here. I'm going to be talking about our NERC-funded project, and it is all around antimicrobial resistance and novel sensor technologies. So obviously, I'm going to focus particularly on the sensor development work that we're doing, and I just want to start by acknowledging all the partners on the project, and so particularly my colleagues at Heriot-Watt University, so Professor Marc Demoulier and Dr. Vasilios Mitrikos, who's the postdoc on the project, and they've been working on the electrochemical sensing that I'm going to talk about, and I'm going to briefly mention some of the work that's being done at IIT Madras on paper-based sensors. So I'm just going to start by a bit of an introduction to the project, basically what the motivation of it was, and I guess all of you obviously know about the problems of antimicrobial resistance, and we heard a bit about it in the last talk. I think what might not be so well known is the role of the environment, and particularly water, in the spread of AMR, and that's what our project is working on. So we want to be able to make new sensors so that we can track the spread of AMR in the water and understand more about that process and how it works. And also what we're quite interested in, so the bacteria in the water will also exposed to other things, so for example the presence of heavy metals or antimicrobial chemicals in the water, so those things are also damaging to bacteria, and they will evolve resistance strategies to those, and then some of those will overlap with antibiotic resistance. So the presence of other chemicals in the water can also drive antimicrobial resistance, and that's something that we're interested in trying to understand in the project, and map how the presence of these different chemicals in a water catchment, how they interact with the emergence and the spread of antimicrobial resistance as well. And we're working with colleagues in India. One of the reasons for that is the scale of the problem there. So India is one of the largest consumers of antibiotics. It's also quite easy to access antibiotics, to purchase them without a prescription. So that's obviously one example of some sort of not so good regulation that's leading to environmental problems of AMR, and then obviously there's other problems around regulations in terms of discharges into the water environment of things like heavy metals and antimicrobials and antibiotics, so from industry and agriculture and hospital waste. So these are the overall objectives of the project, and obviously the two that I'm going to concentrate on talking about today are the sensor ones. So we're looking at making some electrochemical sensors which are focused on detecting the antimicrobial resistance genes themselves. We did think that we would also extend those sensors into detecting some of the co-selectors, but that's probably not something that we'll actually have time to do in the project, and our colleagues in India are focusing on paper-based sensors for the heavy metals and the antimicrobials, so we'll leave that side to them. So first of all, I'm going to talk about the electrochemical sensor work. It's what we're doing at Heriot. So I'm going to talk a bit about the low-cost carbon-based LTCC electrochemical biosensors and how they're manufactured via screen printing, some of the work on the functionalization protocols, and then obviously we're moving into characterization, and we need to identify the right probes to put on these sensors so that we can identify the DNA targets that our Indian collaborators have been finding in the water supplies in India. And then our plan is, once we've produced our sensors, to take them out next spring and test them in Aberdeenshire with our partners at the James Hutton. So we're going to validate the sensors against the existing protocols that they're using in water. So I mentioned there a little bit about kind of what are existing kind of technologies, and we also heard a bit about that in the previous talk as well, about PCR technologies and sequencing, which is what our collaborators in India are doing, obviously sequencing samples from the water that they've collected, which obviously then requires access to that expertise and equipment. In terms of other kind of AMR sensors that are out there, I think a lot of things that I've seen in the literature are a bit more for medical applications rather than environmental applications, and so you're quite different sample matrix, sample volume, and sometimes perhaps the testing might have a bit of a different goal. So some of the sensors are looking at things like bacterial growth, comparing them with and without antibiotics to then enable maybe decision making about what kind of antibiotic to use, whereas we're actually looking for the presence of different genes and how they're driven by these co-selectors. So we want to be able to detect the genes themselves. And the reason that we want sort of sensors for this, it was something that the World Health Organization advocated, that really to kind of tackle this kind of AMR problem in the environment, then what we really need is kind of a uniform approach to surveillance, and that we need more analytical tools that are accessible to low-income countries, so that then we can undertake things like more field testing. There are a wider range of end users who could undertake that field testing, and we can gather more evidence to kind of understand the processes that are happening. We can use them to evaluate the success of different treatment or mitigation strategies, and if regulations are put in place, then obviously sensors can be used to monitor compliance with that sort of thing. So the project, the electrochemical side of things, builds quite a lot on some work that's been published that Professor Marc Demoulier did in collaboration with Till Bachmann. Till's from the University of Edinburgh, and this is the joint article from 2016 is shown on the slide, and that talks a bit about some of the manufacturing functionalization, which I'll also go into some more detail on. The article shows the selection of the type of carbon paste, BQ242, that they selected based on the peak-to-peak separation performance. And then you can also see in the bottom left kind of the schematic, or the kind of principle of operation of the sensing technology. So you have the sensing surface, and the little blue strands are DNAs that are immobilized on the surface. So those are peptide nucleic acids. So they're kind of like a stable DNA analog that is then used to recognize the DNA target. And so that target is the red thing that comes in and hybridizes in the kind of second part of that schematic. And then you can see in the bottom plot how you can then measure the difference between the signal when they're just the probes there and then when the probe is added. And so you have that signal shift and basically an increase in the charge transfer resistance in the system. And then what they've done previously is look at some of the performance of the system, looking at things like what's the concentration of PNA probe that you should use. So and this is really figure A. You can see the measured signal change there when hybridized DNA was added to different concentrations of PNA probe on the surface, and 3 bars on the left are the manufactured LTCC sensors compared to some commercially available DROPS sensors. There's not really a trend in terms of concentration of PNA, but that you can see that the 10 micromolar concentration of the PNA does perform better. So that was then taken forward for utilization to test against a target. And so this was against an antimicrobial resistance gene, BLA NDM. And then you can see basically the curve of response at these different target concentrations, and that the was determined 200 nanomolar. So this was quite reasonable compared to some of the other gold electrode-based systems that have been used before, although they do have a slightly lower limit of detection, and that's something that we're probably going to need to kind of work on improving, especially when we look at the levels of the resistance genes in the water and the kind of concentrations that we need to be able to detect. This slide shows the fabrication process and how these sensors are made. So you've got the base is this low temperature co-fired ceramic substrate, and it's in the laser cutter to the desired size. And then we use screen printing to add the electrodes onto that. And the first stage is to add a silver layer for the reference electrode and interconnects. Then in order to be able to kind of connect up the sensor, we actually add in 5 other layers of the LTC substrate to make it a bit thicker. So we have to bond that together with hydrostatic pressurization and sintering of the layers. Then we take that and we need to add on the working and the counter electrodes, which are made out of the carbon paste. And so obviously there's alignment issues there when that's then screen printed. And then we put a dielectric layer on the top and then dice out the individual sensors from the array that have been kind of produced in one go. And so these are some images of the manufacturing and a few of the kind of challenges that Vasilius has had. So he had some problems of incomplete printout and some delamination and misalignment defects, but he's managed to optimize process parameters to overcome these issues. And then at the bottom, you can see kind of a characterization of our sensor on the right against a drop sensor, and it's behaving well. Then the next stage is the sensor functionalization process. So first of all, there's a synthesis of a diazonium salt in situ, that's then electrografted onto the surface and activated with EDC and NHS for the PNA then to be added on. I think that's actually a repeat, so I'm just going to skip on to the next slide. So this was some data showing the electrografting process, and then where Vasilius is now is we don't know exactly which PNA probes we want to use, so he has kind of a model PNA that he puts onto the sensor surface and the complementary DNA target, and that's seeing like the expected behavior that I showed in the first schematic of the sort of shift, increase in charge transfer resistance when the DNA binds. So that's kind of where we are at the moment. He's been working on optimizing this manufacturing process and the functionalization steps. And one thing that we're finding really challenging is the whole sort of biological side of things. So our Indian collaborators have been doing rounds of sampling in the water in India and generating lots of data from there, and it's sort of translating that information into the best detection strategy. And so they've been looking in surface waters, and so this graph shows abundance of genes in river, lake, and groundwater samples in kind of different areas. So areas where kind of around the source, it's a kind of low residential area or a highly dense residential area where there's a lot of industry, or a low agricultural or high agricultural area, and across kind of lots of these different conditions, the most abundant antimicrobial resistance gene that they found was SIL1. Also been looking at monitoring kind of what happens through sewage treatment plants, and this is one of the ones that they've been monitoring. So they've been taking samples from different stages of the sewage treatment process. And this is some of the data that they'd collected of looking for 40 different antibiotic resistance genes. They've detected 9 and quantified those. And as you kind of go across the graph from the left, so the left side is kind of the incoming influent, and as you go sort of through, this is the kind of the progression of treatment to the final effluent and the final solids. And you kind of see there isn't really much change in kind of the level of these genes, so there isn't really much removal in the treatment steps. And you can see that again for the SIL1 gene, both in kind of high residential and low residential sewage treatment areas. So obviously SIL1 is clearly one of the good, one of the important antimicrobial resistance genes to track, as it seems so abundant in our kind of Indian water samples. Other ones that they have highlighted as the most important ones to look at are PAR-C, BLAR-TM, and Intel-1. So our next stage now is to identify the appropriate PNA sequences from these genes, to then be able to immobilize those onto the sensor and detect the DNA. Yeah, so that's quite challenging. We have like a full sequence data for these genes, but that's a difficult task then to kind of extract out the right information. So we're looking at kind of the PCR target kind of region at the moment, and that's roughly kind of where we are. And so I guess so far I've just talked about kind of the idea of just detecting an antimicrobial resistance gene, whereas actually what we want to do is look for the presence of several different genes. So we want to create a multiplexed system where we can detect these 4 genes in one go. And obviously the system could be modified for other applications or other areas, or to add in different genes by a redesign of PNA probes. So Vasilius is working on a low-cost readout system to be able to basically have a low-cost, easily portable, small system that could read out the sensors, could read out a multiplexed array of the sensors. And so this is his kind of design of how things are going to look with 6 sensors on his device. More of my background is in microfluidics, so this is where I'll kind of come in. Once we know a little bit more about the performance of the sensors and the limit of detection, then we'll know more about the sample processing that we'll need to do. Beforehand, we can design an appropriate kind of sample prep strategy and microfluidic delivery to the sensors. So essentially the sample will come in and be split across these different sensors and then read out by the readout system. I think the slide has also got a little bit messed up, because I think hiding between the 2 graphs is kind of a picture of the prototype system so far. Vasilius has started testing that. CV curves will look how he expects, and in terms of the EIS mode of system testing, says things need to be stabilized a little bit more, but he can at least get the readout of kind of magnitude and the phase of the impedance. He also wants to extend the frequency of that so that it's working at higher frequencies. So yeah, that's kind of where we are on the electrochemical sensors. I'm going to give a brief little overview of what the Indian collaborators are doing in terms of colorimetric paper-based sensors for heavy metals and antimicrobials. So their strategy is to make paper-based sensors, and one of the first things that they looked at doing was a fabrication method. So this is a method just using a kind of normal printer. Therefore, it would be very easily accessible to a lot of people to be able to make devices like this. So you just print out your design, and then there's a heating step to kind of transfer the toner through the depth of the paper. And this has been published in scientific reports last year. And the targets that they're looking at have also come from the different types of water sampling that they've been doing in India, so particularly copper, because they've seen that agricultural areas record high concentrations of copper from sort of pesticides and fertilizers for crops. Residential areas seem to have quite high quantities of lead, which could be due to seepage from pipelines or from paints. And in industrial areas where there's a lot of tannery, obviously the processing of tannery effluents has released lots of quantity of chromium into the water. And they've also been looking at antimicrobials, and the two that they've been looking at are ciprofloxacin and triclosan. And they've also been following kind of how these evolve during a sewage treatment plant. And at least for these, we are seeing removal through the stages, particularly during the maturation ponds step. So basically that kind of the water sampling analysis has kind of led them to select these heavy metals and antimicrobials and identify the kind of concentration ranges to target. And then what they've been doing is adapting kind of existing colorimetric assays and transferring them into paper, and you can kind of see the performance, the colours on the paper and the kind of linear sort of correlation of the concentration and the kind of colour intensity on the paper, and that's for copper. They've done the same for chromium, and you can see there is a difference, that the limit of detection is higher for the paper-based system than using spectroscopy. So there's definitely some improvements to be made in detection, and that's the same for ciprofloxacin as well. So they're trying to explore some different technologies. They're looking at an aptamer approach for the lead to bring things down into the parts per billion detection range. And then a lot of work is looking at kind of different pre-concentration strategies, because a lot of the sensor work there is in the parts per million, but really it'd like to be down in the parts per billion. So they're exploring different pre-concentration techniques, and there's also a link with the University of Edinburgh work in this project, where Edinburgh are looking at the different polymers. Mark Bradley has this kind of polymer array set up where you can screen different polymers for their kind of binding capabilities. So we're looking to see if there are polymers that would be particularly good for the pre-concentration of the antimicrobials. So in summary, the aim of the project is to develop sensors to enable us to detect antimicrobial resistance and its co-selectors in water. We've been working on electrochemical sensors, and we know from previous work that these are capable of detecting antimicrobial resistance genes. And I guess where I've written challenges to select the correct functionalization protocols. I guess I really mean to identify the correct DNA binding probes for this. In terms of paper-based sensors, for co-selectors, these have been fabricated and tested. There's ongoing work to solve the challenge of the limit of detection, which also could be something that we need to investigate more for electrochemical sensors. So thank you very much, and I'd be happy to take any questions.
Thank you very much, Dr. Bridle. That was a really interesting discussion. If anyone has any questions, please. Oh, we've got one right now from Dr. Patel. Hold on one more second.
Hi, Helen.
Hi.
Thank you. Really interesting. And as far as functionalization is concerned, we all who work in biosensing, we know how difficult actually is surface functionalization. And I don't know, how is the type of specificity with the kind of functionalization protocols do you use? Do you target functionalization directly on top of electrodes or in the vicinity of electrodes?
Yeah, it's directly onto the electrodes. So some of the previous work they did, they looked at well, I'm not sure if that's the same as what you're asking actually, but they looked at, once they put the PNA probe on, how specific was it to the DNA target of interest, and they challenged it with different DNA probes and could see good specificity.
Okay. Okay. Perfect.
I actually have one question as well about, sorry. I want to ask, when you're using your electrochemical sensors in those water samples, does the water sample have to be pre-treated? And if so, or if not, I guess, how do you protect your electrochemical sensors from biofouling?
Yeah.
If that is an issue.
Yeah, there definitely is a whole sort of pre-treatment step. So when they will, in India, when they take the water sample and do the traditional method, then they'll start with 100 milliliters of sample, and it obviously can be, depending on where they've got it from, really quite gunky. So there's filtration stages, there's centrifugation steps. One thing that we really need to look at, what would be ideal with these electrochemical sensors, would be that we wouldn't have to do any pre-amplification of the genes. And I think probably for SUL1, if you look at the levels in the water and the levels, like the limits of detection that we can get, then we're probably just about okay. But probably for the others, then we might have to amplify. So then that obviously adds in a whole other, another set of processing that needs to be done. So yeah, we need to look at the whole sort of process and how we can automate some of that. So some other work I've done around sort of waterborne pathogen detection, we've been looking at kind of microfluidic methods of concentration to replace centrifugation, for example. So that might be something that we could then integrate with the sensor technology.
Great. Well, thank you very much. Thanks again, everyone. Thank you. Thank Dr. Helen Bridle for her presentation. I'll move on now to our next speaker, who's another expert in the field of electrochemical biosensing and sensor design, here today to discuss two new biosensor technologies designed to tackle challenges faced by antimicrobial resistance in COVID-19. From the University of Strathclyde, please welcome Dr. Damien Corrigan.
Thank you. Just checking that can you hear me?
Oh, yes, we can. Yes.
Yeah, you can hear me. And can you see my slide as well?
Oh, we still have some slides that are being shared right now. Oh, there we are. Okay. Yep.
Perfect. Okay. Sorry, I'm not the best with technology. Just wanted to check we're running. Yes. So I'm going to talk about electrochemical biosensors for infectious disease, and I'm going to talk about two example projects from our lab. But first of all, I just want to quickly talk about why we would use electrochemical biosensors, and I'll go through this quite quickly. So they can be low cost, they can be highly sensitive. So for antibody-antigen interactions, you can measure in the picogram per mil range, and for DNA, you can achieve femtomolar to picomolar sensitivity. You can multiplex, so you can have several analytes on a chip, and that's really important in sort of modern challenges, because if you think of the best example of the electrochemical biosensor, say the glucose blood monitor, it's a single biomarker. Whereas conditions like antimicrobial resistance and sepsis, they're more complicated, and there is very rarely one catch-all biomarker, and so you need a panel, and electrochemical sensors are ideal for that. They can be applied to a wide range of topics. We tend to focus on health in our group, but as you've just seen, they can be sort of applied to the environment, or to there are other areas like chemical processing, agriculture. So they have a wide range of applications, and they're really there's a lot of potential for integrating with new technologies such as biological printing, integration with smartphones, and microfluidics for sample handling and sample delivery. Now, in our group, we work on a few electrode systems, screen print electrodes, which are low cost. We do some fabrication of microelectrodes and microelectrode arrays. We started to work with printed circuit boards. We started to do a little bit of work with field effect transistors. Now, in the group, we work on a range of disease areas, and in the team we have projects on assay development, device fabrication, but we do some work on molecular biology around isothermal amplification methods, and we think about sensor manufacture. We have projects on epilepsy, endometriosis. We do quite a bit on liquid biopsies for cancer. We have some projects on DNA origami, so functionalizing electrodes with complex DNA structures. What I'm going to focus on today is around AMR. And AMR for us comprises a few topics. So we've done some work on drug-resistant TB. We've done some work on general detection of antimicrobial resistance markers, and some work on sepsis. But today I'm going to talk particularly about antibiotic susceptibility testing. It's this idea of rapidly pinpointing which antibiotic to treat an infection with. And I'm also going to show some results we have on what we've been doing recently on development of a detection methodology for SARS-CoV-2 and COVID-19. So, on antibiotic susceptibility testing, so we've done a lot of work on genetic sequence-based biosensors, and continue to do so. But in starting this project, we wanted to take a slightly different approach, and really what informed our thinking was the idea that classical microbiology techniques still dominate, and when you talk to clinicians, they still trust these techniques, probably more so than the other techniques. They're quite skeptical about some of the sort of biosensor-based technologies or sequencing. And genetic testing identifies the presence of resistance genes, but the bacterial genotype and the phenotype are not always the same thing. So you can detect genetic evidence of resistance, but for example, the right promoter sequences might not be there, or the bacteria might not express that resistance. So identifying a resistance gene doesn't always necessarily equal the AMR phenotype. So we're interested in developing new routes to quick determination of the correct antibiotic to treat a given infection. Clinicians tell us that they would just really quickly like to know which is the right antibiotic. They even would rather know which is the right antibiotic than what the organism is that's causing the infection, if that means they can quickly get the patient on the right antibiotic. So we're working on a technology we call microplate that can handle a variety of sample types, has been designed with mass manufacture in mind, and can be sensitive and specific. So what that looks like at the moment is we've been working with low-cost screen-printed electrodes, and our plan is to move onto sort of more high-end microfabricated arrays. But in essence, the sensor works like this: that you have a 3-electrode system on, at this point, a screen-printed substrate. You then drop cast on agarose, and we've done some work on what electrochemical reporters and nutrients and antibiotics we can include in the agarose without perturbing the electrochemical signal. So when we have a deposit with no antibiotic versus a deposit with antibiotic, we can see growth in the case of no antibiotic and impairment of growth when the antibiotic is present. We can measure that on a potentiostat, and we've done some characterization work of the basic screen printed electrodes involved up to this point. And what that's enabled us to do, first of all, we started off by showing different growth patterns for Staph aureus in this left plot versus MRSA, which is the methicillin-resistant Staph aureus. So we've done some tests with amoxicillin and oxacillin. So these are antibiotics that should impair growth of Staph aureus, but antibiotics which should enable some growth of MRSA. So we measured a range of electrochemical parameters through the gel. We've got these gel sort of functionalized electrodes with then samples of bacterial culture going on. And then we also carried out some MIC assays as a benchmark. So these are just the standard minimum inhibitory concentration tests to see, did our sensor produce similar results to the sort of standard agar plate techniques. So for Staph aureus, we found with amoxicillin at 8 micrograms per mil, a concentration that should prevent growth. We saw no evidence of growth on this, what we call electrochemical growth curve, whereas without amoxicillin, we saw evidence of growth. Now for MRSA, we saw growth with no amoxicillin, similar to the Staph aureus situation. We saw some growth with amoxicillin at 8 micrograms per mil, and then we saw no growth with a very high inhibitory concentration. And we found that these results matched the MIC. And for us as engineers, sort of, kind of sort of simple-minded towards molecular biology, we just thought AMR would be sort of a binary thing, but we found that for, in this case, MRSA, when you have 8 micrograms per mil of antibiotic present, there is still some impairment of growth. So these bacteria, it's not like an on-off situation. It's like they can still grow at an inhibitory concentration for the non-resistant organism. So we found we were able to detect MRSA growth, and we were able to correlate that very closely with the MIC benchmark assays, which is nice. So we've then gone on to enlarge the measurement window. So initially we were just doing this like in an open system. So we've gone on to build what we call a jig or a holder that can handle multiple electrodes. It's made the deposits of the gel a lot more consistent. So here we have a drawing of the gel deposit, and then we can pop on our bacterial culture in here. Then here's the impedance of the gel, and this orange trace is the open system, and then this green trace is multiple electrodes in the closed system. So we're getting a flatter baseline, giving us up to 8 hours here of measurement window, which is important because to detect changes of different species, one species we're interested in is sort of mycobacterial species, things that, for example, things like TB. And so we need a long measurement window to be able to have a chance of measuring slow-growing organisms. So we first applied the system to E. coli and streptomycin as an antibiotic. We can see here in the impedance, the green is the baseline, so we do some baseline electrodes in the system. The blue is when we've added streptomycin at an inhibitory concentration, and then the red is where there's no antibiotic presence. And so we can see a divergence, and from that we're able to infer bacterial growth. We're now starting to work on trying to almost sort of signal processing, I suppose is the best way to call it, but ways of determining growth as early as possible. And we're looking at different electrochemical parameters. So this is the normalized impedance at 100 kilohertz. We've also started to look at the phase angle. And we can see divergence between the two situations, one where the bacteria isn't growing, one where it is. So we're now actually moving on to more advanced signal processing techniques to see if we can deconvolute these signals much earlier. So that work is currently ongoing. So we are in the process with Scottish Enterprise through some high-grade spin-out funding, doing a large number of tests with clinical UTI samples. So we're trying to apply this technology to clinical UTIs. And we're designing a prototype alongside the testing of clinical samples. So we have, for example, an electrode prototype and prototype holder with sample heating and some rudimentary form of sample delivery. That's like an ongoing project where we're trying to make the test more like a prototype, so it could perhaps be tested in a clinical lab or something like that. So now I'm going to talk about some of our work on the electrochemical detection of SARS-CoV-2. So as you all know, we're in the midst of a global pandemic declared back in March. We had this COVID-19 illness caused by SARS-CoV-2, and as you speak, there's greater than a million deaths been recorded. So a very tragic situation, and as Julian was saying earlier, it's really highlighted this requirement for rapid diagnostic testing. So we've sort of entered this domain with our own project, which I'll talk about in a minute. But again, in designing our approach, we've really done this with manufacturing in mind, because in the electrochemical literature, you see all sorts of exotic electrode modifications that give incredibly sensitive surfaces with very admirable electron transfer rates and things like that. But I sort of read those papers and they're great, but I wonder how you would ever manufacture a sensor in that vein. So we've sort of, again, tried to embed manufacturing right from the beginning of the project. So we're working on a CSO project that we're leading, a COVID-19 rapid response call, with NHS Greater Glasgow and Clyde, and then LifeScan, who are a company up in Inverness who make diabetes test strips. So a really interesting company in many ways. They're able in their facility in Inverness to produce around 20 million sensor strips per day for diabetes. They have a very well nailed down, well characterized manufacturing process. We've also been working with Aptima Group, a company who produce commercial aptimers, and Flex Medical Solutions, who are a local sensor design house. And just to remind anyone, here's the basic anatomy of the virus. We have the nucleocapsid protein and the spike protein. The spike protein enables kind of localization on the cell surface and facilitates cell entry, and that will be the main theme of the slides that I'll present in the next few slides. So before I talk about the project, I just wanted to give some background on one of the routes we've chosen to go down. So we've been working with a professor in the chemistry department here at Strathclyde called Glenn Burley, who does some work on the Flores effect. And we've been working on some DNA detection work with him, which sort of, that work has sort of given us the familiarity with the Flores chemistry. So what I mean when I say Flores chemistry is I'm talking about hydrocarbon molecules where, say, for example, a lot of the hydrogens have been replaced with fluorine atoms. And when you start to make these sort of fluorine-containing hydrocarbons, they take on very interesting character. So they become hydrophobic, and they can become lipophobic as well. So here you have a perfluorocarbon, and you can see how water and hydrocarbon are separating out. So we feel that this is a good system, and it enables you to put down a very hydrophobic layer on the surface, and also that hydrophobic layer should have good resistance to biofouling. So we've been working with this molecule called perfluorodecane thiol. So it has a sulfur here, and then it's just a chain of carbons with various fluorines protruding off. Now here we have a diagram of SARS-CoV-2, the spike protein, and how it attaches to ACE2. And ACE2 has this hydrophobic region which embeds in the cell membrane. And that's what we're really trying to do here in this project, rather than sort of putting down a complicated self-assembled monolayer, as many people do in biosensors. We're just trying to form a simple film of this perfluorodecane thiol, so almost making like a synthetic membrane on the electrode surface that would facilitate ACE2 entry and immobilization. So to walk you through some of the results we have, we've been working with PCB-based sensors in this bit of work. We start with the clean gold surface, onto which we immobilize the PFDT, so the perfluorodecane thiol, to make this self-assembling monolayer. So that's just an overnight incubation in PFDT solution. And then we literally spot ACE onto the electrodes and leave it for an hour, and then wash it off. And then that leaves us with a surface where we can do capture of spike proteins. It's a very simple premise. The idea is you functionalize a gold surface, as many manufacturers of diabetes test strips do. They put down a hydrophilic or hydrophobic layer, and then they print on their enzymatic preparation. So our strategy kind of closely mimics the established method for manufacturing diabetes test strips. Once we have the sensor assembled, we do impedance measurements. So we're measuring charge transfer resistance, so the diameter of the semicircle. So to give you some example of what this looks like, here in the case of the clean electrode, we have the black semicircle. Once we put the SAM layer down, the red one, and then the blue one to show the ACE2 being incorporated into the layer. Here's a box plot which just shows that for the individual electrodes on the printed circuit board. When we started off this way, we looked at the SARS-CoV-2 spike protein and streptavidin protein, both of which in these experiments were conjugated to horseradish peroxidase. We wanted to have horseradish peroxidase present so that we could visually test whether the proteins were present on the surface, because HRP is used in ELISA assays, and we've been using TMB, which is a color changing molecule that is produced by HRP, and measuring that as an independent way of verifying and binding. But if you look on the left, we have some impedance plots. So we have the impedance increasing as we titrate in more SARS spike proteins. This is recombinant SARS-CoV-2 spike protein. What we're then able to see here is a comparison of SARS-CoV-2 spike protein binding in the red versus streptavidin protein binding in the blue. And I should say that these streptavidin protein concentrations are actually far higher than the equivalent. So the SARS-CoV-2 spike protein is 1, 10, 50, and 100 nanograms per mil. These are sort of dilutions of a 1 mg per mil streptavidin solution. So these are an order of magnitude more concentrated. So we're getting good discrimination between the spike protein and the streptavidin protein, just to sort of show specificity. We then carried that on by evaluating the spike protein against IL-6. So IL-6 is a signaling molecule which is elevated in people with sepsis and people with COVID-19. So we just wanted to check that we could discriminate between the spike protein and the IL-6. And again, here we have increasing impedance with increasing recombinant spike protein concentration. And again, a similar dose response curve with the LOD being similar. So both experiments giving us an LOD of between 1 and 2 nanograms per mil for the spike protein, with good discrimination against the negative control protein. So we were happy with these results and confident that we were getting good discrimination between IL-6 and streptavidin and the recombinant SARS-CoV-2 spike protein. So we've then moved on to look at samples of SARS-CoV-2 from the molecular standards inactivated virus kit. So this is samples of SARS-CoV-2 that have been grown in cell culture, and then the cells have been lysed. So the positive controls are different concentrations of SARS-CoV-2, and the negatives are basically where there's been no growth of the virus, but it's still a complex cell medium, not unlike so like a saliva sample or a blood sample. But in the case of adding inactivated virus samples to the sensor, again, we get the increasing impedance, we get this increasing dose response curve, we get a slight drop off on our final point, and we're looking into that. We think that could be because these are going through repetitive sort of successive incubations, that ACE2 could be being lost from the layer at this point, or there could be other explanations, but we see good discrimination again between the negative controls and the positive controls. And here these concentrations are given in digital copies per mil. So this inactivated virus kit has been sort of validated by PCR. And so that's giving us very good control over concentration. So the levels of SARS-CoV-2 detected so far in saliva of patients have been between 10 to the 2 and 10 to the 8 digital copies per mil. So we're in the right ballpark. These are being done at the moment with half-an-hour incubations, but we think that we can get the incubation time down. So we're quite encouraged by these results. We think that there's prospects of actually improved sensitivity when we test in clinical samples, which is coming up. And that's because the inactivated virus that we're testing with has been gamma irradiated and heat treated at 65 degrees for half an hour. So we feel like a lot of the spike protein will have been decomposed. So we hope with patient samples, there'll be a lot more intact virus for us to detect. So when we did our limit detection calculation, we sort of fitted and did that. We got a LOD of around 40 digital copies per mil. So I think we have the basis here of a very mass manufacturable, very sensitive assay, and all of this work was run at ambient conditions, so it has potential to be sort of rolled out into real-world use. We then also started doing some work on aptamers with a company called Aptamer Group, who I mentioned earlier. So just for those who are unfamiliar, very quickly, an aptamer is a oligonucleotide sequence which, when folded, takes on a structure, and that structure is very much like an enzyme or an antibody, like it has that recognition capacity that we see in a lot of bioreceptors, and these are at the heart of biosensors. That's the ability to specifically recognize a target. So Aptamer Group have this system where they have a library of single-stranded DNA or RNA. They then introduce the target. They elute off what doesn't bind. They keep what does bind, and then they go through this process of enrichment to end up on sequences that are highly tuned for the target of interest. So they've sent us an experimental aptamer sequence, which is 81 nucleotides long, and an optimer sequence, which is 33 nucleotides long. So this is a shorter sequence, which is where they think the sort of really specific recognition happens within the sequence. So it's from our point of view, from an analytical perspective, this is potentially better because it's a smaller object that we're immobilizing on the electrode. So this work is very much in its infancy, but we've done some basic testing, which I'll show in a minute. But the idea here, so this slide doesn't have animations, but this is a gold electrode provided to us by LifeScan. It has this area where you can connect a passivation layer, and then a working electrode, which is based on a 5 nanometer thick gold surface. This is a very thin gold film. This exposed working electrode. And these are the types of sensors that go in their Vireo range. And it's sort of, again, a sensor very much in keeping with the diabetes test strip. And we've basically been immobilizing the aptamer and the optimer onto these electrodes. And this is just some example results. So we have two cases here. We have the aptamer on the gold surface, and then the spike protein binding. And we've used IL-6 as a control again. And we hope IL-6 doesn't bind to the aptamer. So over here we have spike. Here we have IL-6, both at 80 nanograms per mil. And the red bars are the aptamer. And in this case, we backfilled the electrodes with BSA. The blue bars are the optimer. And again, we've backfilled with BSA. And then, the blue are the optimer, yeah, then the green is an optimer sequence without backfill. And we see, in all cases, actually, that we get good discrimination between positive and negative. The optimer without backfill gives us the highest sensitivity. But we're now, having sort of uncovered promising sensitivity and specificity, with the optimer looking the most sensitive, we're now engaging in tests on the inactivated virus kit to see if we can produce these types of results in a more complex medium. So to conclude, rapid electrochemical antibiotic susceptibility testing has been shown possible for Staph aureus against MRSA with methicillin, oxacillin, and actually amoxicillin modified electrodes. So we've done that with 3 types of antibiotic modified electrodes. By sealing up the antibiotic susceptibility sensor, we've been able to enlarge the measurement window, and that's enabled us to do some tests with E. coli. And now we're doing these UTI-based testing with E. coli samples, UTI samples. We're developing ACE2 and aptamer-based biosensors for SARS-CoV-2. The inactivated virus tests look good for the ACE2 sensor. They remain to be done for the aptamer-based sensor. And then some point soon, we're going to be doing some testing in clinical samples provided to us by our colleagues at the hospital. So what are the main issues we encounter in developing these sensors? So the sensing layer formation and the electrode heterogeneity together can cause high inter-device variability. So what I mean there is forming these self-assembling monolayers is an inherently irreproducible process. So trying to make that as reproducible as possible is a constant theme within our work. And then producing electrodes with low intra-device variability is a challenge as well. So when you combine this process of irreproducible sound formation with sort of poor electrode control, you can end up with very high variation in your analytical data. So getting that variation down by having good quality, consistent electrodes and a really good handle on sand formation, those are challenges that we're always faced with. And as I say here, both of these things can contribute significantly to measurement error. And then finally, as with any biosensor, I think as people have mentioned earlier, moving into complex samples can affect sensor signal. And that's where we think we've got a good advantage, particularly with our perfluorodecane thiol layer, in that we're exploiting that fluorescence interaction. So fluorine wants to self-associate, so it makes these very well-associated, tightly packed SAMs that want to exclude water and want to exclude hydrocarbon. So we think those layers are particularly resistant to biofouling, so that gives us some advantage in measuring in complex samples. So that concludes my talk. It just leaves me to thank members of the group. So many of the guys basically gave up their summers to work on the coronavirus project, and I really appreciate that. It was like a real team effort, and there was lots of negative results that I haven't presented here. We tried various assays with antibodies and things like that and couldn't get them going, but everybody worked really hard on the project. And finally, just to thank some of the clinical and academic collaborators that we worked with. Okay, so that's me finished. If anyone has any questions.
Thank you very much, Dr. Corrigan. Again, yeah, if anyone has any questions, we got one right away. I was warned this would happen. So Kaylee is asking, really interesting talk. Thank you, Damien. What are the main advantages of electrochemical detection of SARS-CoV-2 over technologies currently being explored by other groups, for example, lateral flow? I think there's a multiple part question here.
Yeah, no, I'm looking at that. So from my perspective, the advantage and the reason we kind of conceived and went ahead with the project was that we were trying to tap into the existing manufacturing capacity that exists around diabetes test strips. So the guys up at Inverness can produce 8 billion strips per year. And when you think about the glucose home monitoring kit, it's already CE marked. There are already existing circuits for the reader. The reader's already CE marked. There are very well established manufacturing processes, and people with diabetes buy and utilize these products every day. So we decided to go down the route. And the glucose test is an electrochemical test, and there's a lot of know-how around printing, because that test is based on the glucose oxidase enzyme. There's a lot of prior knowledge about how to stabilize that enzyme, how to print it, what kind of adjuvants are needed to be printed on the sensor to kind of give it long-term storage capacity. So we felt that by trying to tap into an already existing technology that's cheap and highly manufacturable and then has potential for quantification, we thought that kind of gives you a technology that's appealing in many ways. And then also we decided, as a lot of people do genetic biosensors, we decided to go with the ACE2, and we also tried antibodies because of this issue of false positives associated with current tests, and also because of this issue of maybe you're collecting viral fragments that are from dead virus. There's talk in the literature of being able to detect sequences 9 weeks after infection. So we decided to go with the enzymatic detection method because we hope that we'd be detecting live virus as well. And that, coupled with the strong manufacturing capability, we thought that made this quite an appealing approach.
All right. And thank you very much, Dr. Corrigan. We'll have to move on to our next speaker. Thanks again. Closing off the first session for this event, our last speaker, who has worked extensively in the development of multi-sensory point-of-care sensors, is here today to discuss the development of biosensors for the detection of disease biomarkers. So from the University of York, I'd like to welcome Dr. Samadan Patil. I'm asking again, Dr. Patil, are you there?
Sorry, my microphone was mute. I'm here.
Great.
Now, how do I change slides actually?
So there should be a button at the bottom of the screen that has like a left or a right arrow. In this case, it should be the right, so it's the first slide.
Okay. Okay, perfect. So
Great.
that's fine. Perfect. Great. Actually, I'm feeling quite nostalgic for this conference actually, and it is simply because of the Scottish connection of this conference. And another reason is I used to work at University of Glasgow until last year. So it was nice to see some of the Glaswegians over here actually. It was quite nice. So here I'm going to present you different family of sensors. I was quite fortunate to work with different sensors ranging from micro electromechanical systems to magnetic field sensors plus CMOS based sensors. So this is quite a good overview for you from my side. So let us go through a family of sensors or different sensing applications which I accomplished over here. So first I will present you an integrated device which uses two types of sensors. One is MEMS and magnetic sensor. So it is like a hybrid device of two. Then I will cover my work which I did at UCL and in Imperial on development of nanomechanical sensors, again for diagnostic application. And then I will cover my recent work which I did at University of Glasgow on CMOS sensing, and this resulted in one spin-off company, and I will also describe that work. So let us move with the first one, where most of you might have seen MRI machines or electroencephalograms. They're quite bulky because they almost occupy one room, and the gigantic size of any of these machines is because of the squid. So squid coil is used to produce very high magnetic field, and it requires superconducting coil, and that superconducting coil also has to be cooled down by helium or any coolant actually. So could we actually, is it possible to miniaturize this one? This seems quite very daunting task actually. It's quite daunting. And could we possibly integrate some sensors and form a helmet? Or if we could have some handheld device, and that handheld device can be moved over heart, and can it record our sort of a spectrum or magnetic footprint of heart or brain? So that sort of is the strategy over here. So I'm not sure if most of you know that any device, any electronic device we use around us, or even human behavior actually, it follows 1/f noise. So 1/f noise is simply, as the term suggests, if we do something repetitively for human behavior, we keep on reducing the errors. And almost similar thing is true for sensors. If we operate sensors at higher frequency, then noise which is associated with the sensors at 0 frequency or at DC current level, that is absolutely high. And when we operate it at higher frequency, the different magnitudes of the noise, they keep on going. So one of the best candidates for this application was spin wall or tunnel junction. So you can see in this particular figure, there is spin wall and tunnel junction. And if I say that all of you are right now using spin walls and tunnel junction in your computer, you will be surprised actually. So early hard drives, maybe in 90s, they used to use spin walls. So spin walls, the device is very simple. There are two ferromagnetic layers, one top of each other, and they are separated by copper. So the magnetoresistance was around 8 to 15%. So remember this figure, 8 to 15%. And these ferromagnetic layers are separated by copper, which is metal. And the moment you replace this copper with insulator like MgO or other insulator or dielectric, extremely thin, means couple of nanometers only, the figure magnetoresistance jumps from 16% to 400%, and that's the magic. And this happened something around 1998 to 2000, where you might have seen our hard drive capacity went up from, say, 1 or 2 gigs to almost 500 gigabytes now. So this is quite a huge jump, and that jump was simply because hard drives, they started using tunnel junctions instead of spin walls, and that actually increased the reading capability of these sensors. So now these sensors, they have magnetic sensing capability, and they can sense the magnetic field. Just remember this number, 10 to the power minus 6 Tesla, 10 to the power 6, or in micro Tesla we can say. But the magnetic field which comes out of our heart or brain is of the order of picotesla or femtotesla. So imagine there is a long way to go to improve the magnetic field sensing. So one idea was, can we bring one type of sensor, which is MEMS, and spin wall together, marry them, and exploit the combined functionality? Normally things, they don't go well actually in the marriage, but sometimes they do go for physical work. And so in MEMS, actually, if we exploit the functionality of MEMS, that they can actuate it at any frequency. So what we did here, we integrated sorry, I can play animation over here actually. So you can see, I don't know if anyone from the control can move this slide arrows, but anyway. So
unfortunately we can't. Sorry about that.
Okay. So over here, this is the sensor. This is micro electromechanical system, and if this micro paddle moves up and down at higher frequency, then that higher frequency can assist magnetic field which is generated on top, or this is actually permanent magnet or magnetic scavenging layer. So what magnetic scavenging layer does, it brings the magnetic field which is a magnetic field in the vicinity of the sensor. It brings it over and direct it towards the spin wall. So, and if you regulate or modulate this magnetic field at higher frequency, then spin wall can easily see it at higher frequency instead of looking at it at 0 frequency. And that's how the spin wall noise is reduced with the help of micro electromechanical system. So we actually fabricated 3 to 4 generations of this sensor. So if you look at this scanning electron micrograph, this top layer is magnetic flux guide. So it is called COZRNB, quite complicated name. We need not have to go into the pronunciation. And here is the gate electrode, which is used for the actuation. And you can drive this MEMS component at desired frequency. And once this actuates, you can control operational frequency to reduce the noise. So imagine over here, spin wall will have higher noise over here at 0 frequency. As soon as we operate spin wall at higher frequency, this signal which was buried under noise, which is red is noise, the signal which was buried under noise, it stands out very well. So within first design, so this is spin wall, and spin wall is again flanked by 2 magnetic flux guides, which are like butterfly. So we improved from micro Tesla to 500 nano Tesla actually, which was small improvement. But when we drastically changed our design and shifted from spin wall to tunnel junction, so here in this case we changed the magnetoresistive sensor and we removed spin wall and added actually the tunnel junction. So the sensitivity of the device jumped by 2 or 3 orders of the magnitude. So suddenly it came to 40 nano Tesla. Again, when we actually improved on magnetic flux guide from both sides over here. In the last generation of the device, it went into picotesla regime. So imagine we started from almost hundreds of nanotesla, which is in microtesla regime actually, to several hundred picotesla, which is very close to the magnetic field which comes out of heart. And these devices can potentially be now used into a cap or in a handheld device, which can detect at magnetic field signatures coming out of heart and brain. So this was really a daunting task in the beginning, because at the end of fabrication process, either we used to left with only one device, like their bringing together was quite difficult. So first, either MEMS used to there, but spin wall used to vanish, or either way, or even if both were there, they won't work together. Actually, it was quite difficult because they won't communicate. Finally, we managed to get it working, and it was very good success story in terms of device fabrication. Quite one of the most difficult devices I worked so far, actually. Now let us move away from this type of sensors, and we can go towards biomarker sensing. And I'm quite happy in a way that previous presenters or the speakers, they already covered biosensing. So in most of these presentations, you can see that effort is on early detection of the biomarkers. You can see what are biomarkers. Biomarkers are actually the indicators or the molecules which represent particular state of the body. And when you say state of the body, say when you are healthy, then concentration of the biomarkers related to particular disease, they are lowest. So let us say this particular region. And when we are either infected or in a state of some disease or its early onset, then concentration of this biomarker starts increasing. It starts gathering a pace actually. And our aim is to measure these biomarkers actually at this particular stage when person is running and healthy, rather than for the later diagnosis when the person is already hospitalized and he is not capable of working anymore. So aim of any device over here is early detection when the person is up and running, capable of working. So in this direction, the first device I was working on was early detection of HIV infection, and it has been a very massive problem. Imagine a population very similar to half of the London's population being infected every 2 years. So now we are living in pandemic, but even during pandemic, there are other diseases which were actually getting traction before pandemic, like many forms of infection. So HIV was one of these. And this is sort of the schematic of the chip. Here is the inlet, and you can see there are microfillers, and they are for the sorting of biomarkers and removing debris. And these are sensors which are microcantilever sensors. These are very similar to the one which Julian showed in one of his slides. And these microcantilevers are very small micro driving boards which are in a swimming pool. And as you populate the surface of these microcantilevers, they tend to bend, and you have to measure this bending. And this bending is specific to the quantity of biomarker actually, and you get the deflection which is proportional to the biomarker concentration. So this is the microcantilever chip which I fabricated. You can see there are 8 microcantilevers. They are approximately 500 micron long and 100 micron wide. Thickness is 100 micrometer. And these sensors, they were used for sensing of the biomarkers. And as it was mentioned by Helen, actually, she also used quite significant biochemistry, and also the last speaker, he also used quite significant different algorithms for biofunctionalization. So it takes quite huge amount of energy and efforts to get it right actually. And I really appreciate anyone who comes up with very specific biochemistry or surface chemistry for the detection of biomolecules. So here we use gold thiol chemistry, and within gold thiol we use combination of NHS and PEG molecules. And on top of that, we have to use the receptor antibodies for looking at the biomarker proteins for HIV. But instead of using entire antibody, we use the heavy chain fragment, which is called nanobody actually. So we use llama nanobodies for this as a capture or receptor molecule, and those receptor molecules, they were able to capture the genes of interest later on. Okay, so here there was a movie, but sorry, it cannot play since I have files stored somewhere else. Otherwise, there was very nice graphic over here. It shows how a microcantilever, which is functionalized by specific molecules, bend, and the one which is reference cantilever doesn't bend. It was similar to this one. But anyway, we'll just go ahead. So you can see we replicated a microcantilever surface chemistry on other type of sensor, which is called as a plain gold chip or surface plasmon resonance. So surface plasmon resonance is a type of a machine which you get from Biocore, General Electric. So it is from GE Medicine. And then we replicated the exactly same chemistry. The lowest detection which we could see for these biomarkers was around 50 nanomolar. And exactly same chemistry, no change at all. And we could measure down to 500 femtomolar, and this is more than 6 or 7 orders of magnitude higher than what we see by SPR chip. And imagine the massive increase in detection was because of design of the sensor first, and then use of highly specific nanobodies or llama nanobodies, which we use in this particular case. And this actually resulted in quite nice publication. When we were presenting these results in one of the conference of clinicians, a couple of clinicians, they approached us and say, okay, you guys can detect protein biomarkers very well. Could you detect protein biomarkers related to bleeding disorders? So imagine this is called as a royal disease as well. So once you start bleeding with no apparent reason, actually, it could be very ordinary reason that you hit your arm somewhere else, and there would be internal bleeding actually. And this is because of deficiency of factor VIII. So factor VIII proteins are the proteins associated with hemophilia, a disease related to the bleeding. And factor VIII protein is one of the protein in coagulation cascade. So it is the coagulation in our blood happens because of multiple proteins, and factor VIII is one of that. And if we have deficiency, then we suffer profuse bleeding actually. And this was very interesting case for using any microsensors for similar application. Because imagine these patients, they have to go twice to the clinic in a week, and every dose costs them almost 1,000 pounds. And if they take any dose without any requirement, then they are taking actually blind dose, which can increase concentration of factor VIII unnecessarily in their body, and it also costs them or NHS extra 1,000 pounds per week. If you look at the saving NHS can do by in a month is like almost 5,000 pounds, 4,000 to 5,000 pounds, and in a year per patient it will be approximately 50 or 60,000 pounds. So the disease burden which can be saved by using appropriate sensor is massive actually. So you should be able to detect whether you are really having deficiency of factor VIII or not, then only take a dose. And if they go in NHS, normally they get result after 3 days, and actually that test is just for their mental satisfaction, but it is not handy for them for instantaneous dosage. So this disease was quite good model for using any handheld device. You can see that we were able to detect significant, we were able to cover significant dynamic range for these biomarkers, actually, from 1 international unit per mil to 200 international unit per mil, which is very similar to the dynamic range covered by state-of-the-art techniques which are presently used in the laboratory. So most of these equipments are made by Roche, actually. So the collaborators were quite really happy with the results, and this also resulted in one of the spin-out called 3P Sense at UCL. Another work which I did at UCL again was to monitor the efficacy of antibiotics, because not all antibiotics individual we take, they are actually working. So we were working on looking at efficacy of these antibiotics, and we were short of samples. So one of my colleagues, he approached one antibiotics developing company in the United States, and its name was Targanta Pharmaceutical. And they had a new antibiotic called oritavancin, and they didn't know how it works, and they were actually almost denied FDA approval since they didn't have a sort of mechanism of operation of this drug. And they contacted our group, and somehow with trial and error we figured out mechanism of operation of this particular drug, actually. So if we compare this new drug, which is oritavancin, with existing last line of defense antibiotic called vancomycin, you can see the binding pocket is almost similar in both the drugs, but only side group you can see over here is different. Now, this particular antibiotic was tried on 2 types of strands, actually, of bacterium. So one was vancomycin susceptible targets and vancomycin resistant target. And as it was expected, vancomycin targets, they were functionalized on top of these microcantilevers, both susceptible and resistant. So when we passed vancomycin over these sensors, on vancomycin susceptible target, we could see the nice bending. You can see this signal over here within first 5 minutes with the concentration less than 1 micrometer. But for vancomycin resistant, the bacteria which doesn't respond to vancomycin, even if we increase the concentration beyond 300 micromolar, it was not budging. So these microcantilevers, they can look at the sensitivity or efficacy of the drug, actually. And when we use this particular new drug, you can see the slope of operation of this or response of this sensor to the drug. With again 1 order less magnitude or the concentration of the antibiotic, we could see the rapid response for oritavancin of this sensor, actually. And even for vancomycin resistant targets, we could see equally good signal. So imagine for the bacterium which doesn't respond or which is not affected by vancomycin, oritavancin was able to kill that particular bacterium or was able to attach on the target quite efficiently, and we could pick it up very nicely. And you can see that this is the control sensor. It doesn't show any variation. So this was again a very good piece of work which helped the company to get the FDA approval later on. And now I will cover the last part where I worked on University of Glasgow to develop handheld device. So aim of this particular device was to look at multiple biomarkers simultaneously. So far you might have seen that in most of the presentation, there is tendency to look at only one type of biomarker at a time. So here our aim was to look at tens of biomarkers simultaneously. So if you could check 10 biomarkers simultaneously, then their complementary abilities or their complementary detection or you can say that their presence in different states can emphasize particular state of the disease. So for example, for prostate cancer, we picked up these metabolites, and these metabolites are biomarkers for prostate cancer as per the literature actually, because for prostate cancer, the only you can say certified biomarker is a kind of protein actually. And if that particular protein is present, then in blood, then prostate cancer is there. But that particular biomarker test is not also very conclusive, because there are many false positives or false negatives actually present. So we actually used a cohort study and validated this particular technique. I tested this for sepsis, for kidney diseases, and for cardiovascular diseases. So there were four different cases: prostate cancer, sepsis, acute renal failure, and cardiovascular diseases. So this particular sensor, which you are going to see actually now, was validated for at least 3 to 4 conditions with our clinician colleagues. So this particular device is a CMOS chip, and the sensors are integrated on the CMOS chip, and it can be attached to any Android phone or Android tablet. So you don't need massive peripheral electronics for this actually. So just hook it up to your Android phone and you have the operational device, and that was quite handy actually to be used in any location. So this is how the chip looks like. So it is very similar to the chip which most of us we use in mobile phones or in computer chip. The only difference is that every single pixel in this particular chip has 3 types of sensors. One is photodiode, second is ion sensitive field effect transistor, and third is single photon avalanche detector. And if there is a transduction mechanism or molecular visibility which cannot be picked by ISFET, then it will be a photodiode can pick it. Or if there is another physical quantity such as a charge which is invisible by photodiode, then any charge related transduction can be sensed by ISFET. So that was actually the aim, that try to encompass as many transduction mechanism within a single chip as you can, and you can increase the number of sensors. So presently in this particular chip, these are approximately 250 sensors. An array of sensors actually reduces noise and it gives you very good statistical distribution. So the aim was actually to take tens of biomarkers simultaneously, and for that I had to do some post-processing to compartmentalize it into different microwells. So these are different micrographs which shows the creation of microfluidic channels or microwells on top of this particular device or on top of this particular microchip. And these microwells are similar to the microwell plate actually. And each microwell can be used for individual biomarkers later on. So if you could increase number of microwells, you can increase number of biomarkers you can detect simultaneously. So the sensing principle is, from top you shine LED light, then there will be assay done in individual wells, and at the base of every well there were sensors, and those sensors were looking at change in light intensity resulting from color development, and this color development or the rate of color development was monitored as sort of a signal. So this is very similar principle of operation which is used in spectrophotometers or spectrometers, which are routinely used in laboratories actually. So our aim was to condense many spectrometers on a small chips. So these are different biomarkers: choline, sarcosine, and choline and sarcosine, they are routinely used for cancer. Cholesterol and choline, they are for sort of, oh, okay, I am going to finish in 2 minutes. Okay, so these are the results for the cancer. Actually, I cannot show these results over here because they are under review. These results are under review. So for the known samples which are suffering from the prostate cancer were given to us, and we looked at those samples and we picked up the concentration of biomarkers, and that process showed almost 90% validity. So imagine if we blindly check these samples by a handheld device, we were able to predict that the person is suffering from prostate cancer. In this particular device was used again for stroke samples, and we were able to look at the patients suffering from sepsis or kidney function failure, and we could see it actually. So here there was a movie which was able to show that this device works in a couple of minutes. These are the visuals of the device, and this is sort of a final device prototype which we envision as a final to go in a market. This resulted again in a company. We got some funding as well, and there are many companies which are approaching us for the commercialization of this particular technology. So now I go into more details of this device, and I will stop here for the sake of time actually. Thank you very much.
Thank you very much, Dr. Patel. Sorry again about that reminder there, but it was a very interesting discussion about the development and optimization of your devices. We have one question in the chat box here. If I can bring it up.
Let me check.
Apologies for this. Connor asks, hi, great talk, thank you. And I am just wondering about the micro cantilevers. They look quite challenging to fabricate. What were the main challenges involved with that?
Oh, okay, yeah, exactly. So as you already mentioned, they are quite challenging to fabricate. These are the last step of fabrication where you release these micro cantilevers. This is really the most crucial. And this is the final stage. Like you spend almost a month or 15-20 days to fabricate entire wafer. And in a last step, if you make mistake, then all the cantilevers are broken. So it is final releasing step which is most crucial. Yeah, and that is most difficult.
All right, thank you very much. Once again, thanks to all our speakers for this first session of our this year's Future of ISM. Thanks again, Dr. Patel, and thanks again to everyone. Once again, we'll be going into a break before we start session 2. So now's the time where we're encouraging people to go and look at the posters in GatherTown that have been submitted there. We have 3 poster panelists today, and they'll be there in rooms at the top left of the area. Worth noting here is that there are instructions for how to use and navigate GatherTown if you're not familiar with it. You can find that in the proceedings that we have sent to everyone. So please check that out first if you haven't had a chance to yet. Otherwise, you can find them all at the top left of the town. Thanks again, and we'll catch you all in, at 1:30. Thank you.